Well, now on with the program. I would like to invite as a chair of the panel of frontlines of the fight against a global pandemic, the Oxford coronavirus vaccine, the director of Bio-Manguinhos, Maurício Zuma. He has a BS in Business Administration, a PhD in Technology and Innovation from Sussex University in the United Kingdom, and a Master's in Management of Science and Technology from the Public Health School at Fiocruz. He has an Executive MBA in Administration from the Federal University in Rio de Janeiro, and he's an expert in public administration from the Brazilian School of Public Administration at the Getúlio Vargas Foundation. As the keynote speaker on this panel, we would like to invite Professor of Vaccinology at the University of Oxford, Sarah Gilbert, who will be participating online. Her work has been geared to the development of vaccines against influenza virus and viral pathogens, emerging viral pathogens. She led the team to develop the COVID-19 vaccine at the University of Oxford. She is one of the 100 most influential women on the list of the BBC in 2020. As a speaker on this panel, I'd like to invite global health professor in vaccinology, professor in infectious diseases and pediatrics from the University of Oxford, Sue Ann Costa Clemmens. Who will make a presentation, clinical trials of coronavirus vaccine in Brazil. She is an infectologist, a pediatrician, and one of the main researchers in the clinical development of the Oxford AstraZeneca vaccine against COVID-19. She contributed to the development of over 20 vaccines and pharmaceutical products licensed globally. At the University of Siena, she is the director of the Institute of Global Health, as well as senior consultant for the Bill and Melinda Gates Foundation. I'll give the floor now to Dr. Maurício Zuma. Good afternoon, everyone, ladies and gentlemen. It's a pleasure to be back here on this panel. We're getting to the end of the closing ceremony of our symposium. We've had excellent lectures, and we hope to finish with just in the grand style. We're going to have excellent lectures by two distinguished experts. I think this last panel will be extremely useful, and then we'll have the closing ceremony and the awarding of prizes. Vamos começar com...
Sarah, are you hearing us?
I can hear you. Yes, good afternoon. I hope you can hear me.
Yeah.
Thank you very much for inviting me to speak with you this afternoon.
Okay, Sarah.
So I will go ahead and share my slides. And I hope...
Just say something, Sarah. Well, it's always a great pleasure to meet you. You know, every time I meet you, I feel very honored. So thank you very much for accepting the invitation to participate in this symposium, our symposium. And I'm sure everybody here is looking forward to listening to you on the front lines of the fight against the pandemic, the case of the coronavirus Oxford vaccine. Please, the word is yours.
Thank you. So I'm going to talk to you today, and I hope you can now see the slides. Yes, good. I'm going to give you a rather personal view of how I came to be involved on the front lines of the fight against a global pandemic, as is shown here. So this was a situation I never expected to find myself in. I came to Oxford to work on genetics. I ended up working on vaccines against malaria. And then I started working against vaccines against flu and other outbreak pathogens. And then really in 2014, had the first opportunity to contribute to the control of outbreaks using novel vaccines. And this was because in 2014, there was an Ebola outbreak in West Africa, in 3 countries. It started in Guinea, it spread to Sierra Leone and Liberia. Now, this was an unusual outbreak because it was very much larger than all of the ones that had happened previously. It was definitely not the first identification of the Ebola virus. That was first identified in 1976, and there had been many small outbreaks since that time, but most of them had been in remote rural areas. The virus is carried in bats and can be transmitted occasionally from bats to humans. And that tends to happen more in rural areas rather than in towns or cities. And then because the population density is low in these rural areas, there is less opportunity for human-to-human transmission. And once an outbreak has been identified, prior to 2014, it had always been contained relatively simply by using the standard public health measures of contact tracing and quarantine. But in 2014, that didn't happen. The virus spread further, it got into cities, it spread across borders, and the numbers of cases was really beginning to increase very, very rapidly by the middle of 2014. So there weren't at this time any vaccines which were known to be effective against Ebola virus that had been tested in humans, but there were some vaccines in early stages of development or being manufactured in preparation for clinical development. And a group of us at Oxford were asked to take part in testing one of these vaccines. And the reason for that was that it was an adenovirus vectored vaccine, not ChAdOx1, but a ChAd3 vectored vaccine. And the only place in the world where a ChAd3 vectored vaccine had been used in a clinical trial was our clinical trial unit in Oxford. We'd been collaborating with an Italian group who had developed this vector for a hepatitis C vaccine. And so we were asked if we could take on the task of testing the ChAd3 vectored Ebola vaccine and do this as quickly as possible. And we had a lot of support from this. The Wellcome Trust not only funded us to do this work, but they worked with us on the grant application because they wanted to support this work. So we got our funding very much more quickly than we would normally do for an academic research project. We had to get the vaccine put into its vials, which was arranged quickly. We had to get regulatory and ethical approval to do this trial in the UK. But because we'd done a ChAd3 vector trial previously, we had all the documents that we were able to adapt for the Ebola vaccine. We had very good relationship with the regulatory authority in the UK, which is called the MHRA, and with our local ethical committee. And they held special meetings and worked with us to review all the documents as fast as possible. And they did this very quickly. So about a month after asking if we could be involved in this, we were able to start vaccinating. And the first person was vaccinated in September, and by the middle of November, we'd vaccinated 60 people as planned. We had safety data on the vaccine, we had immunogenicity data on the vaccine, and we were able to show that the vaccine was behaving as expected in terms of reactions after vaccination. It was inducing a strong immune response with a single dose. And we were then able to pass this information on to another team in Mali, which was a country next to the outbreak area. They only had a very small number of cases, but thought that the outbreak might spread into their country. And then a second clinical trial was done in Malian healthcare workers, again, only taking a month to do all of that trial. Another 60 people were vaccinated. We were very pleased with ourselves at this point. We felt that we were now going to see this vaccine taken into efficacy testing in the outbreak area, and we would find out if it was going to protect people against Ebola. But that isn't what happened next. What happened was a discussion about the ethical way to test vaccine efficacy when the disease has a very high fatality rate. This was a very important discussion to have, but with hindsight, it was a discussion that should have been held much sooner, so that by January, when trials could have started, there was agreement about how to do this clinical trial. It actually took until April to get agreement on how to do the clinical trial, during which time many people continued to be infected and people continued to die in these 3 countries of Ebola virus infections. By the time the the approval was given to start the efficacy trials. There was a plan to test more than one vaccine, the ChAd3 vectored vaccine that we'd helped work on, and also Merck's VSV vectored Ebola vaccine. But actually case numbers were dropping rapidly in many countries, which clearly was a good thing, but it reduced the ability to test more than one vaccine. And the ChAd3 vectored vaccine didn't ever get tested in these efficacy trials. The VSV vectored vaccine did, which was good news because it turned out to be a highly effective vaccine and could be used. By that time, there were very few cases left. There was little need for the vaccine. And the vaccine also requires ultra low temperature storage, minus 80-degree freezers. The manufacturing process was not scaled up. The vaccine was still being produced at small scale in roller bottles. And this was not ideal for a vaccine to be used in West Africa. But having demonstrated efficacy, there's really little incentive for a manufacturer to go back and revisit the manufacturing process and the thermostability of the vaccine. Subsequently, Johnson & Johnson also produced a vaccine and tested it in non-human primates and showed that in humans they could induce an immune response that matched the protective immune response in non-human primates. And so that vaccine was also given a license for emergency use. But another problem here is that both of these vaccines only cover one version of the Ebola virus, Ebola Zaire, and we have seen many outbreaks of Ebola Sudan virus. And indeed, recently there has been an outbreak of Ebola virus Sudan, and we don't have vaccines to use against it. So for me personally, it was an exciting time being involved in this rapid clinical development of a vaccine, seeing things move very quickly, working with international teams, very collaborative to try to move things forward, but then hitting this bottleneck because one part of the process had not been planned for. And then we didn't see the rapid testing of the vaccine and the ability to roll out that vaccine, which we had hoped to see. But that was a lesson learned for the future. So in Oxford, we've been developing our own adenoviral vectored vaccine, this time ChAdOx1 rather than ChAd3. Adenoviruses are viruses that infect many different species, but instead of using a virus that normally infects humans, we started with one that normally infects chimpanzees. Because with the common human adenoviruses, a lot of people have already had infections with those viruses, and in order to control the infection and rid the body of that adenovirus, they've developed a strong immune response against it, which interferes to a small extent with the ability of that virus to induce a further immune response if you then use it as a vaccine vector. It's not actually a very large effect, but we will prefer to avoid that issue if we can, and that's why we use a simian adenovirus. Adenoviruses commonly cause respiratory infections or infections in the gut. They're usually very mild. We know many people, everybody has been infected by an adenovirus. So it's not a virus that we are not commonly exposed to. And that helps when we have to think about the safety of using a vaccine vector developed from the adenovirus. But we don't want our vaccine to spread through the body and cause an infection after we vaccinate. So we make it replication deficient. We remove the gene, the E1 gene, that allows the adenovirus to make more copies of itself inside a human cell. That gene has to be supplied in a cell line during manufacturing so that we can amplify the adenovirus to make the vaccine. But then after vaccination, the adenovirus will infect cells in the body, express the antigen that we've added, which in this case might be the MERS, the SARS-CoV-2 spike protein, or it could be another viral glycoprotein or a protein from a parasite or bacterium. It will express that in large quantities, but then the vaccine virus does not make more copies of itself. It cannot spread through the body. So even if somebody has a weakened immune system, they are not in danger of a disseminated infection from this virus. And we've been working on this technology for some time. And we've done a number of small first-in-human phase 1 studies using different vaccine antigens. So we had a very good idea of how to manufacture the vaccine, how to store it, what dose to give, what kind of immune response we would expect to see, what the reactions after vaccination were in healthy young adults, at least, which was the population that we've been testing in. More broadly, other simian adenoviruses had been tested in over 6,000 people, and this included babies in a malaria vaccine trial and older people in a flu vaccine trial. So we knew that this was a technology that could be used very broadly. And it's important to remember that if we want to go quickly in making a new vaccine, it's very helpful to be using what we call a platform technology. We have a lot of knowledge about the platform before we start to make the new vaccine. Much of the work has already been done, so we don't have to reinvent the wheel each time. We just have to do the last part of the process, put the new antigen into the viral vector, and it's the same approach for RNA vaccines. And then we can move very quickly through manufacturing the vaccine and getting into clinical development without having to take a very slow approach that we would have to if we were inventing a new type of vaccine every time we wanted to vaccinate against a different organism. So we've been using this viral vector to make vaccines, testing them in clinical trials. But post 2014 Ebola outbreak, it was recognized that the response to that outbreak from the world was poor. We needed to improve. There needed to be more investment, and there needed to be better use of platform technologies so that we were better prepared and able to go faster when we needed new vaccines against these outbreak pathogens. And what happened was that the WHO and a number of other organizations all came up with their lists of priority pathogens, the ones they were most worried about. And these lists were very similar from all the different organizations, and they all included the viruses Middle East Respiratory Syndrome, coronavirus, Lassa virus, and Nipah virus. And at the same time that was happening, the organization known as CEPI was being formed. This is the Coalition for Epidemic Preparedness and Innovation. And this received investment from many different organizations, including many governments from around the world. And CEPI then put out a call for vaccine developers to produce vaccines against MERS, Nipah, and Lassa. And together with Janssen Vaccines, Oxford University applied. And it took a long time to do this application. This was not the same experience that we had with the Ebola outbreak. This took about 18 months to get the funding approved. But finally it was, and we agreed with CEPI and Janssen and Oxford that we would work on these vaccines against these three viral diseases. So let me tell you a little bit more about those. Nipah virus is a very highly pathogenic virus. It's carried in fruit bats, and the viruses can drop contaminated fruit, which sometimes people will pick up and eat, or sometimes animals will pick up and eat and become infected with Nipah virus. It also gets into pots of date palm sap that are being collected, because the fruit bats like to get inside the pots collecting sap so they can drink the sap, and sometimes they contaminate it with Nipah virus. First identified in 1998, so relatively recently, and in humans has caused many small outbreaks with a very high case fatality rate. It can be up to 100% in some outbreaks. As shown on the slide here, you can see the number of cases just in one country, in Bangladesh, which has had most of the human infections. There has been a big public health campaign to try to improve the safety of date palm sap, and that has resulted in improvements in some years, but then in another year, cases will come back again. The virus is not extremely transmissible between people, but it can transmit from person to person, and this typically happens when somebody is caring for somebody else who has a Nipah virus infection, so family members and healthcare workers are at risk. In Bangladesh, they now have very good surveillance teams. They identify these cases very quickly and manage to quarantine those infected. But there are no vaccines and there is no specific treatment. It's just supportive therapy. The concern is that, as happened with Ebola in 2014, there could be a larger outbreak. It could happen in a country which is not as well primed to look out for it as Bangladesh is. Because these fruit bats actually fly over very wide areas, so many countries are potentially at risk. And we don't have any vaccines against Nipah virus, but there is a vaccine against a very closely related virus called Hendra virus. Now, this is one that was found after it infected horses in Australia, again carried by bats, contaminating the pasture where the horses are grazing. horses became infected, seriously ill, and people then in contact with the horses contracted Hendra virus from the horses. The way to deal with this was to develop a veterinary vaccine to protect the horses against Hendra virus. This was registered in 2015. And since that time, when horses in the area that is at risk from Hendra virus infection have been vaccinated, there have been no more human cases because humans are no longer at risk of exposure to horses that have Hendra viruses. Unfortunately, we can't vaccinate all the bats that carry Nipah virus, but because we know that a very effective vaccine has been used against a very close relative of Nipah virus, there are very strong prospects for developing an effective vaccine against Nipah virus. And using our ChAdOx1 technology, we've shown that in non-human primates, we get protection after a single dose of the ChAdOx1 Nipah vaccine. And this vaccine is now in clinical trials in the UK, and we're in discussions with ICDDRB in Bangladesh to continue clinical development in the country that has the most cases. So this project was paused during the pandemic, but is now back on course, and we hope to be able to demonstrate in non-human primate studies, again, that the vaccine is effective, and then to get conditional approval for use in outbreak situations, and possibly to protect the healthcare workers and the surveillance workers who are most at risk in Bangladesh of coming in contact with Nipah virus. Another virus that we're working on a vaccine for is Lassa fever virus, this time found in West Africa. This one is carried in rodents, these small multi-mammate mice. They like to get into people's houses and they will contaminate food stores with Lassa virus because the rodents can shed Lassa virus for many months. And then people become infected when they are exposed to infected dust from the rodents have contaminated. The whole of West Africa is at risk. Most of the cases are in Nigeria. And although infections in children may not be highly fatal, about 10% of children are left with permanent deafness after an infection. And this causes another long-term health problem. It's also extremely dangerous in pregnant women. We've been, again, working on a ChAdOx1 vectored vaccine against Lassa fever virus, again, protective in non-human primates. And this is now being manufactured in readiness for clinical trials, which we will start in Oxford and then, as quickly as possible, move into further clinical development in West Africa, the area that's affected by this virus. But one we've been working on for longer was Middle East respiratory syndrome. We had UK funding prior to the formation of CEPI. This is a coronavirus. It's now endemic in camels in the Middle East, although that hasn't been the case for very many decades. But people who are exposed to camels can be infected with MERS. And we now know that some of those infections, if the person is young and healthy, they can be very mild or asymptomatic infections, but they can still pass that infection on to other people because this is somewhat better being transmitted between people. There's an outbreak reported just this week in a hospital in Saudi Arabia. There have been quite a number of hospital outbreaks in Saudi Arabia and one very large outbreak in South Korea, which was extremely expensive to deal with. It involved 4 hospitals. So we've been working on a ChAdOx1 vectored vaccine against MERS. We took the major surface antigen, which is the spike protein. We put it into our ChAdOx1 vector. We manufactured it and we started our clinical development. So before 2020, when we had to work out how to deal with another coronavirus infection, we already had data showing that a single dose of ChAdOx1 would produce antibody responses against the spike protein from one dose, and that these were well maintained even a year after vaccination. We induced T-cell responses and we induced neutralizing antibody responses. This was subsequently tested in a further clinical trial in Saudi Arabia, again in healthy young adults, and it's now in a further trial in the UK in older people, which is the main target group for this population. It tends to be older people with some degree of compromised immune system that are most at risk of severe infections and hospitalization. But knowing that we had this information on the design of the antigen, the way we put the vaccine together, and knowing that this was producing strong immune responses in people meant that we had a head start when it came to developing a vaccine against SARS-CoV-2, as it became known, and that wasn't a name that we had at the beginning of 2020 when we first started to look at cases of respiratory infection, and then discovered that it was actually a coronavirus. It was a coronavirus that hadn't ever been identified before, and it was starting to spread very rapidly. So I first heard about this on the very beginning of January, the first day of January 2020, started following the story with my small team at the time, decided that we should start making a vaccine even if we didn't know it was going to be needed, but then it became clear that it would be needed. So the team expanded, we raised money, we got into manufacturing. And as Rino Rapparoli was telling you in the last talk, by April of 2020, we were in our phase 1 studies with the SARS-CoV-2 vaccine. Those trials expanded around the UK. They then began in Brazil, which was a very important part of the clinical development, which I think you'll hear more about in the next talk. And then by November of 2020, we had our vaccine efficacy data going on to authorization for use in the UK, and then ultimately authorization in many other countries as well. So the first data that we had from our clinical trials was in July, and we published this very, very quickly. We showed that from a single dose, we could induce neutralizing antibodies against SARS-CoV-2, and that's what you can see in these boxes on the left here. But even in the first trial, we had a small group of just 10 people that we gave a second dose of the vaccine to after 4 weeks to test if that would increase the immune response. The problem at that time in developing a vaccine against a novel coronavirus is that we don't have any vaccines against any other coronaviruses that infect humans. So we really don't have any knowledge of how strong that immune response needs to be. So when we saw that a second dose of the vaccine increased the immune response, we decided that the rest of the trials had to be 2 doses rather than 1 dose, because this would give us a stronger immune response, and that was more likely to be protective. And that's what we then just changed the trials to go from a 1-dose trial to a 2-dose trial. That actually gave us quite a lot of problems with vaccine supply. We didn't have very large-scale manufacturing ready to go at the beginning of 2020. We had to expand our manufacturing. AstraZeneca joined us, and they did an amazing job of setting up manufacturing facilities around the world, and Fiocruz were extremely important in this endeavour. But we did struggle for vaccine supply during our clinical trials. We sometimes had to wait, and that resulted in sometimes having a different interval between the first dose and the second dose. And when we eventually got our efficacy data, you can see here on the column in the right-hand side that we have different numbers for vaccine efficacy for different parts of the trial. And it became clear after some further analysis that this related to the interval between the first dose and the second dose. So if people had had a 12-week interval rather than the 4-week interval, they had a stronger immune response, and those were the people who had the higher vaccine efficacy in this group of clinical trials. We also saw that analysing this at different dose intervals, the longer the dose interval, the higher the vaccine efficacy, and also the higher the immune response. And then we looked at the vaccine efficacy after just 1 dose, and we saw that at least for 3 months after the first dose, we didn't see any drop in vaccine efficacy. This meant that the way the vaccine would be used was to give the first dose of vaccine and then wait 3 months before giving the second, because the first dose would give a high level of protection against the vaccine, and then the second would maintain that protection. But in the situation where supply is limited, and now commercial supply of the vaccine was needed and supplies were increasing, but not as quickly as everybody would have liked, being able to give more people the first dose of vaccine meant that more people were protected quickly, and then they came back after 3 months and got their second dose. So that became the policy in the UK, to give 2 doses 3 months apart, and this was also changed for the policy for the RNA vaccines to give those 3 months apart as well, for the same reasons, even though that hadn't been tested in the clinical trials. And here you can see our chief investigator of the UK trials, Professor Sir Andrew Pollard, receiving his vaccine on the first day that the vaccine was licensed for use in the UK. So what about all those different numbers in the efficacy trials? Well, it's quite confusing having lots of different numbers. But what we have to remember is that it depends on the question that we ask, as well as the way that we deliver the vaccines. So from our trial, our UK trials, we were able to see that there was only low protection against asymptomatic infection, although there clearly was some. This was determined by asking the volunteers to send in nasal swabs every week. So sometimes we knew they were infected, even though they didn't. We had a higher degree of protection against mild illness and 100% against severe illness and death. But when you're designing a clinical trial, we're normally looking at protection against mild illness, and the exact definition of that mild illness can vary in different clinical trial protocols, and that can have quite an effect on the efficacy that you measure. So you can only compare the efficacy of one vaccine to another vaccine if it's been in the same clinical trial, in the same population, at the same time, and is assessed head to head. So what we're really asking here is this vaccine effective enough to use? Should we be using this to vaccinate the population? And the answer from the clinical trials was yes. And then what we need to see is when that vaccine is rolled out and used in the population, what is the vaccine effectiveness? And we were very pleased to see this data set, which was released on the 1st of March 2021. And the policy in the UK was to vaccinate the over 80s first and then move on to the over 70s and the over 60s. And what we were able to see was that in the over 80s, hospitalisation was reduced by 80% after a single dose of either the Chalox or the Pfizer-BioNTech vaccine. This was amazing news. We saw that in the over 70s, again, one dose, we were getting protection against symptomatic disease, not just hospitalisation, and deaths in the over 80s who had been vaccinated were reducing faster than in the younger age groups who hadn't. And over time, more data accrued, and then we went on to second doses. But this is the information that we really want to be looking at. It's, does the vaccine work to protect the whole population, rather than worrying about the exact numbers that are coming out of clinical trials? So supply for the world was really important. And when Oxford originally talked to AstraZeneca about being our partners, manufacturing the vaccine, firstly, it would not be for profit, and it had to be made accessible to as many countries as possible. And AstraZeneca approached this by setting up manufacturing sites at many different places in the world, including at Fiocruz in Brazil, to manufacture many doses, and we ended up with a total of more than 3 billion doses. This was made available largely through the COVAX program. It was the most commonly used vaccine in low and middle income countries for a very long time because it was available, it was low cost, and it didn't require frozen storage, and all of those contributed to it being used extremely widely. And for that reason, in the first year of use, it's estimated that this vaccine saved more lives than any other because so many doses were used in so many different countries. Following the pandemic, in Oxford, we formed the Pandemic Sciences Institute. This is a picture of Professor Sir Peter Horby, who ran the recovery trial, which tested drugs to use against in hospitalized COVID patients. And they very quickly showed that steroids were effective and how to use the steroids to save lives. They went on to test many other drugs. They showed which drugs did not work as well as which ones did. And they made all this information public very quickly. And that was also responsible for saving many lives around the world. But Oxford also had other work done during the pandemic. And as we started to come out of the pandemic, we decided that we wanted to keep everybody working together, all of these different disciplines, to be better prepared for future pandemics. And so we formed the Pandemic Sciences Institute, which is now working to put us in a better position to counter future pandemics, not just through vaccines and drugs, but also thinking about ethics, about how to design clinical trials, all of the things that we found we were missing in those previous episodes that I told you about. We're trying to work with partners to make sure we're prepared across the board, and we're including looking at vaccine confidence and making sure that people are prepared to take the vaccines that we've worked so hard to develop and to test. We are planning a new building. This is an artist impression. We hope to have this building ready for us to move into, along with another group, Jenner Institute, that work on vaccines against different diseases. This is still just an artist impression at this stage. We still need to raise some more money to finish the requirements for putting this building up. And we are about to hold our second International Pandemic Sciences Conference. We did this last year in Oxford, very multidisciplinary, attendees from many different countries, and we were very happy with the outcomes. And so we're going to do it again this year. So if you're interested in pandemic sciences, you can either attend in person, or registration is free for the online conference. So you can use the QR code here, or go to the website and get free registration for this conference without having to leave your desk if you would like to join us. So we need to stay on the ball. There are many pathogens that we already knew about before the pandemic that can cause outbreaks. We need to be much better prepared, and we need to be thinking about the different families of viruses that can cause outbreaks. A big problem in 2020 was that the pandemic was caused by a coronavirus, and we don't know how to make a vaccine against a coronavirus because we don't already have any vaccines against coronaviruses. If it had been influenza, we would have had a better idea of where to start. But we now have to look at the different families of viruses, the filoviruses, the bunyaviruses, the henipaviruses, and make sure that we understand what is required to produce a protective vaccine against each of these viral families. That will mean that we will have vaccines stockpiled ready to use in outbreaks, but also if a new virus is identified, it's likely to be one of the members of these families, and we can then, as we did with the MERS vaccine informing the design of the COVID vaccine, we can use the vaccines that we've already made to tell us the best approach to making vaccines against new viruses that are close relatives. So there's a lot of work to do, but I do think that if we continue to do this and work with all of our partners around the world, we can be better prepared for future outbreaks and future pandemics. So I'm going to stop there and just leave you with my thanks to all of those who took part in our clinical trials in all of the different countries that they happened in, to AstraZeneca, our amazing manufacturing partners, the team in Oxford, the teams in Brazil and South Africa, where other clinical trials took place, and of course our funders, without which we wouldn't have been able to do this work. Unfortunately, we were able to get the funding quickly in 2020 when we really needed it, so that we could get on and make a vaccine and then share it with everybody else. Thank you.
Thank you very much, Sarah. Thanks for this amazing presentation and for sharing your experience in the trajectory of the viral vector platform. It's very instructive for us, all of us. I was wondering that there are several talks across the world on preparedness. And maybe preparedness is to know how you were prepared 4 or 5 years ago. Thank you very much. Let's pass now to Suen. Suen, obrigado por aceitar também nosso convite.
Thanks for accepting our invitation, Suen. Those of you who know her, she is a Brazilian with an international career, and she will be providing continuity to our work. Suen will talk about a topic that's the continuation of the work, all the work that was done at Oxford University. Suen, please, you have the floor. She'll be talking about clinical studies on the vaccine. Good afternoon, everyone. I'd like to thank the organizing committee, but mostly Dr. Akira. It's a difficult task to be the last speaker, but anyway, I've been invited to talk about clinical studies on COVID vaccines, and I have two goals. One of them is to really talk about studies and the hardships we faced and specificities, and number two, to talk about the learning and the legacy, what happened during the pandemic, but mostly as how we, the researchers, industry, government, scientific community, will be working to transform these actions and can make them real for the next to face the next pandemics. It is undeniable that we have had an impact by the pandemic on various sectors, but mostly on the development of products, and among them the vaccines and products, and the development of products from point to point, from beginning to end, at the different sectors in different countries. It took part in different phases on the development of a product, but it's important mostly for the next pandemics that we should be ready for to develop new vaccines and products and diagnostics and tests from beginning to end in all stages of the process. When we began to develop vaccines up until 1925, we produced one vaccine every 50 years, and that has changed dramatically. In 2020, we produced more than one vaccine in less than a year, and that changed the paradigm completely because many phases were combined, because the world was not ready not only to have many products but also to test these products. And once these products got to the population, several developed countries were not ready to do mass vaccination, and mostly to deal with pharmacosurveillance, and that raised actually concerns and led to the anti-vax movement. Among the many aspects that changed, that produced this paradigm shift, as Professor Sarah Gilbert said, from discovery to licensing, it took us a year, and that really changed the paradigm of vaccine development from end to beginning to end completely. Advancement of new technologies, new platforms, as Dr. Rina Rafali said, several platforms that had already come into existence had not been used to that end. Vaccines like RNA messenger, RNA that was used in relation to cancer, for example, and among the platforms themselves, RNA messenger, we had its own evolution, RNA messenger, modified RNA, self-amplifying, and so on and so forth. The fast track, regulatory fast track, on the first day, we had the lecture by Dr. Mehruz, very important lecture. Had it not been for the product is developed, but if we don't license the product and this product doesn't get to the population, we won't have any impact whatsoever on public health. So the continuous submission is very important. In addition, as was said before, the development in parallel of many phases in general, the industry, academic circles develop a product and they wait for results for that phase, and then they move on to the next phase. In this case, that didn't happen. The phases ran parallel to each other. What about the quality? People were asking. Quality didn't suffer. And we showed that this evolution in the methodology is possible, and also to bring safe products to the population. What happened that was different during the pandemic? It was investment, public-private investment of significant risk and an anticipation. There is, as in research and infrastructure as well as in manufacturing, and that was not done before. As I said before, it was one phase after another in the sequence. A product that had not had its efficacy proven, it wouldn't have been produced on a large scale like the case in the COVID. In the rush for vaccines, Brazil made huge contributions. Focusing on our country, it contributed in bringing to the world 2 vaccines in 2020 for emergency use at the international level, as Dr. Sarah said, the Oxford AstraZeneca vaccine and the Pfizer vaccine. In addition, it contributed to the approval of efficacy, to ascertain the efficacy of 5 vaccines. I worked on phase 3 and I conducted studies on 2 of those vaccines. In an ideal world, we have various aspects to think about in clinical research, and we have time and we have time to provide capacity building to the teams and scientists. I try to allocate in two different areas: investigators, researchers, and qualified centers. And in the country, what do we think when we develop a product and we start the global development project? We think of a country with a laboratory that will be able to produce clinical trial material. There is a CRO. There are regulatory times, acceptable schedules that won't hold back the clinical development. And as for researchers and centers, well, I'm not going to go into detail, but the curriculum of that scientist should at least have a 25 to 50% of its personnel already trained in clinical development with a great deal of expertise in that area. And there should be other departments that will contribute to the formation of a center of excellence. Obviously, we're thinking of other things as well, like epidemiology in relation to the product that we're developing, and ethnic and economic diversity, among other factors. Ethnic diversity, inclusivity. The pandemic left us with little time. The world was in lockdown. The air traffic was scarce, and health professionals and in general help us develop the new clinical research. We're on the front lines of the pandemic, so there was a scarcity of qualified personnel and insufficient centers with the proper qualification. During the studies on the Oxford vaccine, many people that were recruited volunteers and many some of my centers were called on by CROs to work at the CROs. So the lack of qualified personnel was acute. It was quite a hurdle to overcome. Given the scenario, many institutions had leadership, exerted leadership, and they sought to prepare centers to welcome phase 3 studies. And that was good. I'm glad the world and scientists and producers were able to get there with many to take several molecules to phase 3, but there were not enough centers. The Bill Melinda Gates Foundation invested billions of dollars in this project that we call site readiness. And the idea was to really work on by region. I got Latin America, and we were able to identify and map out 22 centers and qualify, train, and equip. And in 4 months, in the course of the pandemic, we provided capacity building to over 600 people. But interestingly enough, we wanted centers that could adapt easily to the situation of the pandemic, centers that would have quick recruitment and high quality recruitment with regulatory fast track, capable of increasing resources, human resources and infrastructure that is training, but most importantly, the physical venue, the physical space, because of what was in because of the requirement placed upon the centers, surveillance and hygienization and distancing, social distancing. That was the outcome. 22 centers. We had 2 publications in 7 countries in Latin America in 4 months. These centers were ready to recruit about 1,000 volunteers per month. So through this set of resources, we could recruit 17 to 2,000 volunteers a month. Brazil had 6 centers. At the end of these 4 months, 21 of these centers were either conducting research or negotiating research to be started, to be commenced. This is a center that we worked at an earlier phase. The Institute of André Chagas Institute in Belém, we launched the Center for Pharmacological Products later on, now focusing on the efficacy studies in Brazil during the pandemic. Several projects were placed as a priority by the World Health Organization. They said it would be great if we could, in 90 days, start a line of research here in Brazil, due to the generosity of entrepreneurs like the Redditor and the Lehman Foundation. We were able not only to foment, but also train, equip, and start the studies in 44 days.
And this was against the increasing in cases. And it was important for us to initiate the studies with the increasing of the cases so that we could help and bring hope to the world. We would prove the efficacy of the vaccines more rapidly. In Brazil, we had 6 centers that was distributed along the Brazilian coast, and that would also enable us to have a diversity, social, economic, and ethnic. We were selected by 5 regulatory agencies, and we didn't have any problem. And this shows the quality of the process and the speed of the recruitment. Simultaneously, 19 centers recruited the same number of volunteers, and in Brazil, they went into the efficacy phase around 11,000, but we got up to 12,000. But so we carried out the same work and 6 center in the same simultaneously in 6 center, and so we showed that we were able, and we showed that we had to improve the before and the after, but clinical trial execution was very well carried out. So considering Brazil and Brazilian level, we had prepared in 4 weeks, and we had the CRIO Unifesp, where we show this warehouse, which was full of hospital material, and we transformed it in 4 weeks in the centers that most recruited in Brazil. This was the center that most had the most recruits, and we transformed the basketball court into a center. So we had 17 subjects, and it led this research and also Jansen's research with the variants that we had, and also cover and also the booster studies. However, the registration and the vaccine and the development is not concluded when you get to the end of the process. This keeps on. We have to show the real world evidence, and we have to show these. And we conducted this in Botucatu, and with the municipality governments and all the health secretariat, we were able to vaccinate 68,000 people in 10 hours. So we generated evidence for booster doses, and we can see the mayor of the city and also the UK ambassador participating in the campaign. In the Botucatu study, we made social action. We collected 10 tons of food, and another real-world evidence study that we carried out, led by Fiocruz, was in the Maré complex, and we took these 110 tons of food and we distributed in this study. So we not only learned how to improve this logistics, because recruiting 68,000 people in 10 hours, it's not an easy task, but we were also were able to donate to this community. So we also participated in booster studies and generating evidence for immunization policies, and also the regulatory aspects. As I've said, in the implementation of the study, we participated in the contribution, and regarding the regulatory framework, we accomplished a very important role, and until then we were not within this regulatory sphere in a way that was attractive to international bodies. So after the pandemic, we cannot go back to the previous scenario. So in my group, we invited Mr. Bahatores and his team, Oxford, and he was received in Oxford and also by CEP to discuss the future of the guidelines and regulatory guidelines post-pandemic. We cannot, Brazil cannot leave this international scenario. So this slide was shown by Dr. Sarah Gilbert, and we once more we have to evaluate the impacts of everything that we do. And once more, the vaccines where we contribute to prove their efficacy are among those that more saved most lives. And other important data is that Brazil contributed with more than 7% among all the COVID studies worldwide. So this is very relevant, and we cannot stop here. We are creating training courses in the graduation course, undergraduation course, because the training is very important for the clinical research centers. And this historic begins much in the past, and Latin America has been contributed in the vaccine field worldwide. So we have 233 vaccines that are pre-qualified by WHO, and in 2024 we contributed with 24 vaccines in a total of 27 diseases, with more than 6,000 participants in the clinical trials. I could not refrain from mentioning the clinical trials that we did. I was leading with GSK, but also Dr. Penny Heiden in Merck. We got we received an award for this, and we are not starting today. The thing is the maintenance of these centers and all this ecosystem. I thought that this was one of the most hard studies that I led before COVID, because recruiting more than 10,000 children per month was not easy. However, talking about the future and our legacy and our lessons learned, we had several papers that were presented and several studies, and one of them is undeniable. Then we have to discuss in the G20 summit. We have to have global leadership for these aspects. Number two, we have to talk about funding, and otherwise we won't get anywhere. And once more, coming back to our sphere on the left, you can see the clinical trial centers that are prepared to use. I had to prepare with my group the PAF, and this is also prepared in Middle East, Asia, and Africa to receive phase 3 studies. And what is going to happen with these centers in 2-3 months or 1 year? These centers were going to lose the people, and if they are not trained, and the equipment are going to be obsolete, and once more we are not going to be prepared for other situations. So within this sphere of center maintenance, we have to talk about how to retain the personnel, qualified personnel, and to bring to our regions more teams for phase 1. And we have to have interesting and attractive centers for phase 1. So we have to develop mechanisms to maintain a network of centers that are functioning, and we call them warm centers. In addition, a network of laboratories that can work with clinical trial assays and also samples. We do not have this network, and this is costly, and it affects our budget, and it excludes us from the international scenario. In our case here, all the samples were sent to United States and Europe. And why do we not have this network of laboratories here? Number two, CDMOs, so production of clinical trial materials. Of course, we have in Fiocruz and other institutions, but we do not have this in large scale, and we have to have this. We have two CDMOs that are being launched right now, one in Unesp and another with Bio-Optimize, and that are private and are helping us to get out of the current situation. We talked about G20 and G7. I had the honor of participating as an expert to present to the G7 leaders in a document where we discussed this that we have discussed till now. And we had to have less red tape on this process. When you have all this processes are happening, we have to have a globalized action, but also the unnecessary disbureaucratization. And I would like to quote Larry Brilliant, and he says, epidemics are not evitable. We cannot avoidable, but pandemics are optional. So thank you very much. So congratulations, Swain. Excellent. To share with us the experience that you have in this last years. And Bio-Manguinhos is trying to prepare itself to get into the circuit. We have a clinical trials area that is advancing, and also our pilot plant is being prepared so that we can contribute in the clinical batches. And so we are very sensitive to all this, everything that you're bringing. So we are very tight in time. I'm not sure if Sarah wants. Are you still there, Sarah?
Oh, thank you. We know you are 4 hours ahead of us. Thank you for staying. I have just one question for you, okay? And my question is, can we predict what will cause the next pandemic and where it will start?
I don't think we can predict that with any certainty. We can have our favorites. We're worried at the moment about H5N1 influenza causing infections now in cattle in the United States, and only one human infection so far, but it's very worrying that large numbers of mammals have been infected, because that's giving the virus the opportunity to mutate and adapt to mammalian transmission. So that could be what causes the next pandemic. But on the other hand, we could be taken by surprise again, as we were in 2020. So we can try to have the best preparation for the things that are most likely to happen, but we don't know what country they'll come from, and we may be wrong in our predictions. So I think we also have to be prepared for the unexpected and make plans to deal with lots of different scenarios.
Well, great. Thank you very much. And just one question for Sue. Sue, como que a gente pode criar e manter uma
How
rede?
can we create and keep an international network that is active and prepared for a next sanitary emergency? Thank you very much for your question. It's very relevant. It's a dream of all researchers to have a warm site prepared for this and how to do so. I believe that we need a consortium with international institutions for fostering governments, the industry, academia, so that we can map countries and center and create these centers of excellence. Some of them will be improved, better equipped, but the main issue is to bring the studies, because these centers have sequential studies, and what eliminates the center is having this interval with no activity at all, that we call in development the valley of death. So if you have these overqualified centers, with people that are qualified and they do not have any studies happening. I see this with my students in Africa. They end up opening a drugstore or working at the local bank. And when we see those people who are qualified for this function, which is not easy, because doing a clinical trial is not a matter of only recruiting, but we have to meet all the regulatory requirements so that we can have a report that is submitted and approved. So this person is working at a local bank or a drugstore to survive. So it's not exactly the case of the Brazilian centers, but I've seen this happening in several centers in Brazil where we have to relive the team and centers that became obsolete. And also the Evandro Chagas Center. I carried out a rotavirus study there, and I went back there, and they do not have roof over their heads. So it's very sad to see a center that can produce a lot and be in this state. So the institutions and the actors collaborate. So fostering institutions, funding institutions, governments, private sectors, and they should keep on nurturing these centers so that when we need and we have the next pandemic happening, that we have a place for shelter. So thank you very much, Sue, and
Obrigado a todos. Estamos finalizando o nosso painel. Sarah, can you stay for just 1 minute? Just 1 minute for a picture. Let's take a picture in front of the panel with you.
Parabéns, obrigada a todas.
Thank you, Sarah. Hope to see you soon.
Congratulations. We'll see you soon. Bye-bye. Goodbye.